All right, good morning, everyone. So today we're going to continue our discussion about this idea of balancing metals as far as their importance in terms of different molecular mechanisms, but also we will talk about regulating metals in a way that protects them from them. And so we're going to look at a specific articles that talk about these concepts. I believe we'll have time to discuss two of the articles today. I actually brought in a guest speaker, a student from the course last semester who was very passionate about topics that concern the brain, the neurochemistry. And so she helped me to develop a lecture that we're going to repeat today that's going to really focus in on the importance of treating or addressing the issue of bipolar disease. And the article that I've selected talks about the use of lithium for certain bipolar disease conditions. Right? So it's not actually meant to as indicative of lithium being something that's a cure-all for that particular condition. There is actually no cure-all for that one. All right, so what we're going to do, first of all, I'm going to let you guys know that we will have assignment number 3 posted later today. I'll send it to you by email. A reminder that attendance during the seminar presentations is required. I do record your attendance, but I do evaluate your participation, so that's one of the components that's a key to your success in this course. And so let me go ahead and project the lecture today. All right, so as I've asked you guys to please post, if you wrote talking points for the lithium paper, to please post one of your talking points into the chat, and we'll try to address as many of them as possible. All right, so we're continuing our discussion of this idea of balancing metals in the body. And so here I post again this set of points that I mentioned in our last discussion, and we talked about an example, a case study that looked at the situation in B, where there is a breakdown in the internal homeostatic environment, and so how the body operates in an aberrant manner, characteristic of a disease state. What we're going to focus on today are two case studies that look at these following two topics. The idea that sometimes the body intentionally disrupts the metal-ion equilibrium in a defensive manner, and also looking at how potentially non-essential metal ions can be employed to help restore balance to disrupted processes, but that their levels must be tightly regulated. All right, so let's really quickly tackle the topic of nutritional immunity. And this is one that I have alluded to previously, and it was one of the questions in your second assignment. And so this particular article focuses on how essentially trace minerals, so in this context, focusing on certain transition metals, are regulated to protect the host against pathogenic bacteria. All right, so let's look at what nutritional immunity basically is. It is a process by which a host organism employs molecular species to sequester essential elements in a defense mechanism against infection. And so when you're exploring this form of immunity, you actually have to look at it from the context of the host setting up a defense mechanism, but also the agent that is serving as an infection will pose its own counter defense mechanism. All right, so you see that both the host and the pathogen have their molecular mechanisms in place in order to facilitate metal sequestration. And you can think about this for the pathogen as a way of mobilizing the metals for their own purposes, for their nutrition. So here are the things that, at least in the context of the article that I had you guys look at as some insight into how our body employs certain biomolecules for the purposes of nutritional immunity. So humans employ peptides and proteins to increase retention of essential metals and to minimize the export. So as an example, we have a transport protein located in the cell membrane, ferroportin, that gets downregulated in order to minimize the amount of export of iron when we become infected by, let's say, a pathogenic agent. But then we have the pathogens able to set up their own counter defense mechanism, employing their own mechanisms to do so. So, for instance, bacteria have produced small molecule chelators. So we talked about this very briefly. Siderophores that are extremely important in terms of binding iron, and so they employ metal binding moieties that favor very high affinity for iron. There's also the zinc equivalent of siderophores, so zincophores, that have the capability of binding zinc. And so essentially, these are produced by bacteria, and there are also other organisms that do the same for the purposes of trying to extract metal, potentially from biomolecules that bind the metal. Bacteria have also evolved receptors to capture metal-bound proteins, so proteins that are not native to them, or to decompose proteins that are not native to them but that have a metal-bound moiety. So here's just a representation of the variety of mechanisms that bacteria have developed in order to, in this context, exploit iron-containing agents. And so here we have the representation of gram-positive and also gram-negative bacteria, how they have evolved these mechanisms in order to be able to capture iron-bound biomolecules. So in the context of the gram-positive bacteria, there are certain bacteria that have the capacity to actually recognize hemoglobin. So as you know, that's an iron-containing protein. We depend on it for transporting oxygen to be able to breathe. So there are receptors that have been developed to be able to capture them. There are also receptors that have been developed to capture just the heme component. So that then has to involve a proteolytic digestion of the protein to be able to release the heme group, right? The heme, still having a very high affinity for the iron, and then that gets uptaken into the bacteria. And you have the depictions of small molecules that are not necessarily at the membrane, that they get exported out, and then they can capture heme, they can capture just released iron, and then they get, we have them, we don't have, the bacteria have the mechanisms then to be able to bind these agents and then bring in the iron. All right, so you can see that from this illustration over here on the right, you see some additional features. For instance, there's evidence for certain gram-negative bacteria having the capacity to even recognize the iron-bound transference. So here you have the capturing of that protein, and then that protein is able to get shuttled into these bacteria. So you see these features of sort of the sophisticated machinery that has been built in order for capturing this metal and utilizing it. But that's not the only way in which bacteria are able to really take advantage of the essential metals that keep a host alive. So here's an illustration of the capacity of a bacteria infecting cells, human cells as an example. And so here you see that they can then release siderophores, and those siderophores have a very high iron binding affinity, which can essentially extract iron from transferrin, or even extract iron from ferritin. As you know, ferritin is a very insoluble form of the metal. So that protein is basically a machine that produces nanoparticular iron, but nonetheless, the bacteria have the capability of binding that iron, mobilizing it, and making it so that it is bioavailable. So these are different ways in which bacteria have evolved their sort of counter defense to try to take that iron away from its host. All right, so what are certain additional things that we can learn from this concept of nutritional immunity? Well, metal levels in humans may serve as an indicator of susceptibility to infection. For instance, people suffering from iron overload diseases or requiring multiple blood transfusions may be at high risk for infection. And so the iron overload diseases, that's obvious. There's just too much iron and that iron is available. And so if something, you get infected, that iron is accessible for a pathogenic bacteria to be able to thrive, to proliferate. But to just remind you guys, the issue with blood transfusions, our bodies don't necessarily have the capability of handling excess iron. As I mentioned to you previously, we do have the mechanisms in place for handling slight shifts in the homeostatic control of iron. So we have the ability to control when there's a little too little iron or a little bit too much iron. But when we have a large excess, our body simply does not have the capability of handling that. And so blood transfusions result in an excess amount of iron getting into our system. And so that, and just the nature of how these transfusions are administered can be a big source of infection. Another really important topic that gets brought up when evaluating nutritional immunity is this idea of the relative metal levels in blood. And so there are examples of certain populations that have, relative to what is considered the average amount of certain essential metal levels in blood, to be on the lower end. But perhaps for those particular populations, that may not actually be low in the sense of possibly inducing a disease condition. It may be something that is particular to the geographical location, perhaps because of endemic infectious diseases. And so the body responds to that environment by maintaining a certain lower than normal amount of metals to protect the system. So normal amount of levels within the blood, as an example. All right, so another really important thing to consider is this idea of low metal levels during a microbe infection, and whether or not to supplement someone who is actually dealing with that infection, because the reverse may happen, where by supplementing with the low level metal, you might actually induce disease infection, and it could be quite detrimental. All right, so these are things that nutritional immunity helps us to get oriented about with respect to this idea of the homeostatic control of a metal, and how we have the ability to not just regulate the levels, but also to regulate the storage of a metal and the retention when in the presence of a pathogenic agent. And so from these type of studies, new antibiotic therapeutic strategies have evolved. For instance, you have those sideromycin Trojan horse type strategies, essentially the use of siderophores, which would be recognized by bacteria, but linked to an antibiotic. So it's basically, you have the component that would be recognized by the bacteria, which has the capacity to bind iron, but in the same boat, you're bringing in an antibiotic which will be able to attack the bacteria. You also have probiotics, which is essentially taking advantage of certain bacterial strains for our own defense. So those bacterial strains may not necessarily pose a major risk to us, but they have the capacity to attack highly pathogenic ones. All right, so these are new therapeutic insights that have been obtained from an understanding of what constitutes nutritional immunity. All right, any questions that you guys had with regards to this topic? All right, so no questions. Okay, so what we're going to do, I'm actually going to skip over case study 3. We're going to focus on it later on. What I want to do, let's go skip over this. I want to focus on the lithium subject article. And so what we're going to look at here is how there is metal regulation with respect to protein phosphorylation, but focusing on lithium in the human body. So there is a big question mark about whether lithium is an essential metal or whether it's a therapeutic metal. All right. So in the context in which I'm presenting it to you today, we're going to look at it from the perspective of its therapeutic value. And so what I want to do is just talk a little bit about lithium in the body, and how we do have the mechanisms for being able to transport it and even uptake it into cells. So lithium dietary deprivation studies in animals demonstrate that it can lead to a decline in fertility, which suggests that it may be essential for certain animals. So one of the ways that you can define something as being essential is by designing an experiment in which you test whether the removing a certain element from a diet results in a physiological effect that is potentially detrimental, but then when returning that element into the diet will then be able to help you to recover from that physiological response. All right. So that is one traditional approach that people have done in order to gauge whether something is potentially essential. And so that is what has been done in certain instances to look at whether lithium might actually have some sort of biological importance. Lithium levels in blood plasma are almost directly proportional to lithium ingestion. In other words, the amount that you take in leads to an increase in the amount that you have present in your blood, right? So that may not be so obvious, but there are certain things that you may ingest and it may not necessarily lead to a significant increase in your bloodstream because you have perhaps the ability to process that, let's say, a given metal and be able to excrete it without it actually entering into your bloodstream. All right, so due to its ubiquity, and so ubiquity means how highly abundant it is in our environment, lithium ingested in trace amounts is unavoidable. So you can't avoid having lithium into your system. All right, that's also very true for the metal titanium. Titanium is so abundant in white paint and dust particles that you have a certain amount in your body, and it simply would be very difficult to study that in a context where you have basically a complete depletion of these metals. It turns out that the human body can tolerate up to 1.2 millimolar of lithium in blood. But then above that threshold, that's where toxicity occurs. So you have the following symptoms: cramps, vomiting and diarrhea, kidney dysfunction, coarse tremor, confusion and irritability. Then above a threshold of 3 millimolar, then you have very severe conditions: seizures, coma, and even death. So lithium has the capability of crossing through biological membranes. There is, at least as of yet, no known transporter that has been I can say evolved in order to specifically transport lithium. All right, so there's no, I would say, specific transport mechanism for this metal. However, it has the capability of exploiting transport mechanisms that are specific for other metal ions, for instance, sodium and magnesium 2+. And so here in this table, what I'm trying to show you is a comparison between the ionic radii and even potentially compounds, coordination numbers, not necessarily an actual visual of what they would look like, for lithium with respect to sodium and with respect to magnesium 2+. And so focusing on the numbers that are right here, you'll see that lithium compares quite favorably across the board with magnesium 2+ than it does with sodium plus. Okay, but nonetheless, lithium does have the capability of exploiting sodium pathways. And so here, what I'm showing you is a visual of lithium being able to take advantage of many transporters channels that exist for the movement of sodium and other related metals. Calcium there, you also see potassium. All right, so one major pathway for lithium entry into cells is through sodium channels of the voltage-gated class and non-voltage-gated class epithelial class. So lithium can mimic some, but not all, of the sodium regulatory roles, but simply not as effectively. Lithium efflux, that means so departure from sodium regulatory roles, but simply not as effectively. Lithium efflux, that means so its departure from cells is also sodium dependent. Lithium can transport across epithelial and non-epithelial cell membranes and across mitochondrial membranes. As I mentioned to you, lithium can compete with magnesium 2+, very similar profile in terms of being able to mimic the coordination or the ability to bind molecules as magnesium 2+, but also it's true for calcium 2+, for specific sites in phosphatidylcholine, phosphatidylglycerol membranes. A source of lithium toxicity is its competition with sodium transport. So lithium is excreted by the kidney, and then sodium depletion due to extreme sweating, diarrhea, vomiting, dehydration, use of diuretic medication, or even severely salt-restricted diets can lead to a toxic level of lithium. And the reason for this is because the lithium can be reabsorbed from the kidney tubules instead of sodium, using the sodium phosphate co-transporter in the nephron proximal tubule. And so because of that reabsorption, what would end up happening is that you would end up having a much higher lithium level than expected. So essentially, lithium using these sodium transport pathways can be a source of its toxicity. Another thing is with respect to magnesium is that it can inhibit magnesium-dependent enzyme, partly because it has the capability for substitution of magnesium 2+. For instance, this inositol monophosphatase cofactor. It can also target biphosphate 3' nucleotidase. Lithium inhibition of glycogen synthase kinase 3 beta is believed to be central to its therapeutic effect for bipolar disorders. So that's a very important enzyme, this GSK3B. So what has been observed is that the lithium can bind to an essential magnesium 2 binding site within this enzyme, may be invoked in affecting the activity of magnesium ATP, which is important for the functionality of this enzyme, and may even affect phosphorylation levels of important proteins. And so I mentioned this this enzyme because it actually plays a critical role in the article that I had you guys read about the importance of lithium with respect to the bipolar disease. And just to familiarize you, this is something you would have learned in biochemistry. So just to remind you that the protein phosphorylation process is facilitated by protein kinases. All right, so often this is coupled with a the involvement of ATP. So essentially, the hydrolysis of that molecule resulting in the addition of a phosphate group onto a protein, and then the reverse, the dephosphorylation process, is facilitated by phosphatases. Essentially, these enzymes that can then remove the phosphate groups from proteins. And so I mentioned this because phosphorylation becomes a critical component in the article that we focused on. And so here, I want to show you something that is very busy, as you can see in this diagram, but it's an interaction diagram that has been developed for that enzyme, GSK3B. This was done as a bioinformatics study. And so you can see here I've highlighted, there you have that enzyme, all of the proteins that may be correlated with, in some way or another, with that particular enzyme. Right, so I emphasize this because that interactivity can prove really critical in terms of some of the effects that we're going to talk about. All right, so as I mentioned to you, I have a guest speaker. So this is a Gesmaria Cabrera Rivera, who was a student in the course last year. So she was very interested in focusing on the effect of metals in the brain. And so she was particularly motivated by this subject matter. So she developed this lecture, which complements the things that we're going to talk about in the article that you guys read. So just to remind you, for anyone who wrote talking points for the lithium article, please post one of your talking points into the chat, all right? Because that's going to serve as some additional discussion points. And along the way, we will be addressing some of the questions that I've seen you guys post up. All right, so Getsamari, are you ready?
Yes, sir.
All right then, so go ahead.
Okay, hi guys. My name is, as the professor said, Getsamari Cabrera. I had a wonderful time taking this course, and I hope that you guys are doing too. Please, next. Okay, so we're going to talk about bipolar disorders, but we have to understand that bipolar disorders is a part of the affective disorders, which that means that we have depression, mania, and the combining of these two are what we know as bipolar disorder. Sorry for the background noise. I have a construction next door, so bear with me on this one. Okay, so we know that mood disorders are a very common form of mental illness. Back in the day, Hippocrates was the first one to describe it, calling it melancholia. And later on in history, people recognized it as a disorder of a brain function. It's very characterized by extreme exaggeration of moods, meaning that you can be super happy or, very hyperactive, or you can be really, really sad, but it's not in the way that we see people or talk about it in like saying, oh, I'm feeling depressed because something happened. It's not like that. Um, it's actually, we can't confuse the reactive depression, which is the one that I was trying to describe, with major depression. Reactive depression is just that something happened and you're feeling sad, but is something momentaneously, meaning that you can get over it by a week or two. If you keep feeling sad, saying, for example, after 2 weeks, maybe 3 weeks or longer, maybe 6 months, that's when you have to go to a doctor because you may be having a major depression episode. We have to point out that also there's a cyclical environment, meaning that moods can swing between mania and depression episodes. So keep in mind that not only you can be depressed, but you can also be in a mania episode, meaning that you can be very, very happy for 6 months after being depressed, and then you go back to feeling depressed for another 6 months. Next, please. So, the bipolar depression, or as we know it, bipolar disorder, is a dysphoric mood, meaning that it's a loss of interest with almost everything. You have the inability to experience anything pleasurable, meaning adenonia, and you can have feelings like guilt, crying, desperation, loss of appetite, um, agitation, or motor retardation, meaning that you're moving more slowly or moving faster than usual. You can have localized pain, which it's not caused by strain of exercise or that you were hit in some place. You just have uh, out of the blue a pain that you can't bear with. You have a low self-esteem. Um, you also keep yourself down with self-devaluation, and you have also severe digestive disturbances. This is because there is the vagus nerve, which is really connected with the stomach. That is another chapter. So just giving you that, I think that it will suffice. Next, please. There's also the mania factor, as I said. This part, it's people feeling elation, you feel grandiose, you feel that you feel powerful, you have bursts of energy, you feel full of fun. You don't feel actually the need to sleep. You think that you can be 48 hours, 72 hours awake. um, you are more talkative than usual, like you are talking as a parrot, as we say. um, you have a lot of thoughts and ideas, and you can't actually pinpoint them. Like you start, I don't know if you guys have experienced when you are writing something that a lot of ideas come into mind, like making like a brainstorm, but it's like having a brainstorm inside a brainstorm inside a brainstorm. But you also have the irritability, and you become impatient. Yeah, and I can say that it does causes a headache, and you don't know how to get rid of it. Next, please. So I saw that somebody asked what types of bipolar disorder we have mainly two. We have the bipolar disorder 1, and inside of that, we have over 20 diagnoses that are in the ICD code. Most of them uh, talk about mainly mania or mainly depression or maybe hypomania, which is like you're not in full mania. It's not a full mania disorder. And you can also have them with or without psychotic features, and it's also labeled by progression. You can have a severe, mild, or a low a low uh, feelness of the disorder. Then in bipolar 2, as in the bipolar 1, we have hypomania, which is not a full-blown case of mania, but you also have depression. So it's really considered like a depression slash hypomania. You don't have to really have been in a mania disorder. You do have to, though, be inside a 2 year of this cycle of being between hypomania and depression. And then the cyclothymic disorder, which is another type of another subtype of the bipolar 2 disorder. It characterizes by being hypomaniac, the depression, and you also have 2 years of persistent symptomatology, meaning that it's kind of like throughout your life, and you notice that you can shake it off. But the cyclothymic disorder is distinguished because the symptoms do not meet the diagnostic criteria that we're going to see later on. Next, please. As I said, we were going to see the diagnostics criteria. These are the major depressive disorders diagnostic criteria written in the DC-5 manual. This is the book that it's used by psychiatrists to diagnose somebody. For example, the patient describes depressed mood most of the day, nearly every day. Or in children, it can be they feel irritable most every day or every day. They feel like they don't have an interest. You can actually lose or gain weight without dieting. You have an insomnia or hyperinsomnia every day. Psychomotor agitation. We talked about having a lot of bursts energy, and this is the opposite of that. You feel energy. I'm sorry, the psychomotor agitation. It's like you feel agitated, but this being between psychomotor agitation and fatigue is like you want to hit somebody, but you really don't have the need of doing it. I don't know if you guys are understanding this. It's, for example, you feel really, really down, and somebody intrudes your room, and you want to kick them out, but you really don't have the energy to say it, and you feel restless in the bed. All the symptoms can cause impairment in social in your life. It comes you can be strained from your social circle, and it can also have affects your work schedule also. and important, this is not a comorbidity of or a side effects from drugs that you're taking or another psychological disorder that you may have. Next, please. Opposite to the depressive disorder, the maniac episode, like I said before, you feel powerful. You don't need the sleep. You can come very irritable, and you can actually hit people because you feel this grandiose uh, way. So you feel that you're invincible. Um, a mood disturbance is enough for you to feel away from social areas because you can be an impairment. Um, and also the episode, it's not attributed to side effects of medicines or another event. It just like it can happen out of the blue. Next, please. We have seen in this graph, we can see that I don't know if I have, Professor, can you point the laser? Um,
Yeah, let me see if I can find the. Sorry. Yeah, because right now I have the pencil on.
okay, that's fine.
Yeah,
Can
I can.
you draw?
Yeah.
Can you draw a line um, in the highest points of the lines, please? Okay, we can see that throughout from right to the left. If you analyze the graph, we can see that in the early 20th century, people were diagnosed mostly in their 70s. And as the time progressed, we came closer to being diagnosed at 15, meaning that the depression rates, the rates for the diagnosing of depressions, BPD disorders have been retreating as the people become younger and younger, which is a huge thing to watch because that is a very important detail. Bipolar disorder can be traced as a genetic disorder. So we can see that we have the succession of the two world wars. You can see them that, especially the Second World War, between 25 and 35 years old, they were a really high point for that. And the after wars also, the later wars that came of that also. Next, please. Okay, somebody asked for the neurotransmitters involved. There are three mainly involved. We have serotonin, which is a neurotransmitter that is this is, you feel elated. We can see in this picture that we have the mechanism of how is moved between the neurons. As you can see, we have the tryptophan is synthesized, and it's what in the graph it's called 5-HT, which enters vesicles, and those vesicles are the ones that liberate the neurotransmitter. And then in the postsynaptic cell, we have the receptors. This also happens with most of the transmitters, and they each have their own receptors and vesicles. In the next slide, we have dopamine and norepinephrine. They are both catecholamines, which involve epinephrine and adrenaline. They actually, dopamine and norepinephrine, they have one difference, and it's the alcohol that you see, the after the, yes, that one, sorry. And it's just one more reaction that is done by an enzyme, but dopamine, it's actually the precursor of norepinephrine. In the left, we have the same picture that we saw earlier, but in the postsynaptic cell, which is the one that receives the neurotransmitter, we can see that they are coupled, and they're coupled proteins, which mean that they can inhibit or excite the adenyl cyclase, which can produce or inhibit ATP. Norepinephrine is also similar to this mechanism. The difference is that the receptor, instead of being named D, it's actually alpha, but it has the same mechanism. Next, please. The limbic system in the brain, I wanted to show that these are the parts of the brain that are involved in the neurotransmitters pathway in the brain. We can see that there are very important parts here. We have the hippocampus, the olfactory bulb, and all of those parts are very critical for learning, memory, emotional responses, which is why I brought it, since BPD is an affective disorder, and motivations. And in this slide, we see the main pathways, of the norepinephrine and serotonin. And if we put these two graphs inside one each other, we can actually see that all of these pathways almost go along, like similar in a parallel sort of type. They are very, as the professor pointed out, they have the overlapping nature, and they do interact between those neurotransmitters. And actually, since it's norepinephrine, we can actually extrapolate it to all the catecholamines, which involve also dopamine. Next slide, please. Here we see the different types of the cells. We have a normal cell in the far left, and we see it as a normal survival cell and growth. But when we have stress, that means that we gain glucocorticoids, and we lower the levels of the BDNF, which are the brain-derived neurotrophic factors, which are proteins that act as a neuron growth factor, and they influence the neural growth, the cell differentiation, and their survival. So that is why it is, if we compare it to the normal cell, that shrinks the cell. This causes an atrophy and decreases the survival of the cell, meaning that increases their vulnerability. When we have antidepressants, we actually try to gain the reception of norepinephrine and serotonin, and we also increase the BDNF factors, meaning that we try to expand that cell that it was shrinking, and we try to increase their survival and their growth. In the right, we have pictures of how these cells are seen in real life with their synaptic vesicles, and in the far right, we have the dendrites, which are the which are the I'm sorry, which are the kind of like trees that we see in the cells drawn in the far left. Next, please. Okay, the lithium treatment in bipolar disorders has no effect on the mood or behavior in healthy patients. It reduces approximately 60 to 80% of manic episodes without causing sedation or depression, which is really good because that way we can only focus on the depression. It also reduces the recurrence of manic episodes in the future. It reduces the suicide potential in bipolar individuals. This also makes the hospital stays less, meaning that the patient stays less than 2 weeks, instead of being 8 to 13 weeks. It has been used also to identify bipolar disorders in people if they respond to the treatment, but this is a limited approach because some people do not react to lithium. And it also enhances the serotonin actions. It elevates the tryptophan, which is the precursor of serotonin. It increases the serotonin, meaning that it increases the serotonin by itself, the metabolite, and its release, which alters the receptor response in several brain areas, as we saw in the pathways. Next, please. This treatment reduces the norepinephrine activity by enhancing the reuptake and reducing the release, meaning that it lets the norepinephrine stay in the little space that it is in between the presynaptic and postsynaptic cells. It has pronounced effect in the adenylyl cyclase and the phosphoinositide cyclase, the G protein coupling, and the brain VNDF factors, and other multiple intercellular cascades, as we saw in the receptors picture with dopamine, because it is a very similar to dopamine. It regulates the GSK3B, which was the protein that the professor was talking. That the biological clock regulates that protein, but that protein in parts feeds the modulation of circadian rhythms, which is the rhythm that we use for sleeping and waking up. That is why some people feel very alert that they don't have to sleep. Lithium may enable normalizing this rhythm and sorry, making it the mood stabilizing effect that it has. It also alters the intracellular actions regardless the triggering of the neurotransmitters. This may explain its effect in both mania and depression. As the professor said, lithium levels can be very toxic and they have to be monitored closely. And the problem with this treatment is that its therapeutic index is very low because the levels that are needed to achieve the treatment are very high. So the patients do endure several lifelong side effects can be with thyroids. I can't recall the other ones, but it's a very it has a specific list, and only about 50 to 60% of patients show a good response to lithium. So that is like half of the population that are in this treatment, but nonetheless, it's a good treatment to have. It's a treatment that it's actually one of the first treatments that it was discovered for uh, bipolar disorders. And then we have the second generation of antipsychotics, which are also used in bipolar disorders, and the third generation after analyzing the side effects and all that. So people that aren't treated with the lithium treatment are treated with these this type of treatments also. And I think that is it. Any questions?
Oh, thank you, Gizmari. Yeah, any specific questions for her?
Una pregunta, profe. Una persona que sea diagnosticada con uno de los tipos de trastorno bipolar, ¿podría con el tiempo, o no sé si hay algún efecto que cambie en el paciente, que el diagnóstico pueda variar de uno hacia otro? O sea, de un tipo hacia el otro, no sé si...
La realidad es que el diagnóstico de bipolaridad, como va a estar oscilando entre episodios de mania y depresión, hay que estar bien pendiente. Es un diagnóstico que va a estar por el resto de la vida, así que es...
Sí, entiendo que el diagnosticado, o sea, el diagnóstico de trastorno bipolar es considerado como una causa de discapacidad, ¿no?
Sí.
Por eso lo explico ahí, que es como la sexta causa de discapacidad.
Sí.
Podría, o sea, yo entiendo que uno de los problemas que también existe con estos tipos de pacientes es que ellos también tienden como a suspender los tratamientos. Bueno, entiendo que es por de pronto los efectos secundarios que ellos presentan. De pronto esto con el tiempo puede generarles a ellos algún tipo, no sé, de cambio o variación en el diagnóstico como tal.
Realmente el suspender los medicamentos sí les va a causar efectos secundarios, pero es bien importante, obviamente todo medicamento que se utiliza en psiquiatría no se puede dejar de la noche a la mañana porque los efectos secundarios son bien fuertes y bien percibidos. Si el paciente está consciente de cuáles son los efectos secundarios, los puede identificar. o una persona que esté velando por su bien, pues los puede identificar rápido y a veces son al instante. Algo que quería mencionar es que la bipolaridad puede ser un desorden que puede ser inducido por drogas, otros medicamentos, y también es una comorbilidad, con un diagnóstico de comorbidad con la esquizofrenia. Así que es un diagnóstico que está a través de diferentes etapas y es un diagnóstico que hay que estar bien pendiente porque, como dije, no es solamente depresión o solamente mania, sino que es una oscilación entre esos dos y a veces la gente no se da cuenta. No sé si eso te contesta la pregunta.
Yeah, it is a complexity of diseases, which, then you have lithium, which has its own particular toxicities, which can complex it even more. So I know there was a student who brought up this issue that could lead to possibly an onset of another condition. And it's not clear whether that is the case, but, you're trying to balance one set of processes, and there are multiple things that are getting affected, right? And while trying to do that, yet this metal is inducing its own potential side effects because, as I had mentioned to you, the levels of that metal are really hard to control because your body can reabsorb from the kidneys, and so it's not necessarily easy to get a handle of how much you're actually putting into your body and how much your body is retaining, and then what's actually the steady state. So it's a complicated one, and as Getsamari had mentioned, it's not as if there are many people that have multiple mental health conditions, right? So it's very hard to, and even the diagnosis is not straightforward either. Like the identifying whether someone is lithium responsive, that's its own set of studies itself. Yeah,
exactly. And it's the therapeutic index can, since it's very low by itself and it's really close to the toxicity levels, that by itself, it's a really big risk in taking it. Because not every, like if you are a patient that doesn't respond to the lithium, you are practically poisoning yourself in a kind of way, and you have to catch it before you do poison yourself, because you don't want to die trying to solve one issue and die for another cause,
exactly. This whole idea of regulating the levels of even the treatment here.
It's really. I'm sorry.
No, go ahead.
It's really a very difficult equilibrium to keep, because it also can be treated, with the food that you take. Tryptophan and I forgot the other one. The we can call them the reactives to dopamine, norepinephrine, and serotonin are actually found in food. So if you ingest food, a certain amount of food to keep some levels of those reactive available in your body, you might not have to you may have a better chances of feeling better, but it's not all for sure, since this is an imbalance of the chemicals, you're trying to make them equilibrate again.
And that's quite difficult. Yeah.
So your diet is somehow affected, or not affecting, but could help you possibly to control the symptoms or worsen them?
Definitely. The thing is that if you're in the depressed mood, you don't want to eat. You have cravings, and not those cravings not necessarily have healthy meanings. For example, you want chocolate, which is serotonin, which has the serotonin precursor. But if you eat a lot of chocolate, or if you eat only chocolate, what's gonna happen? You can develop diabetes, for example. If you and then you don't eat meat, well, that's gonna have another problem, because since it's an imbalance of two neurotransmitters, one is going up and the other is going, I'm sorry, one is going up, the other is going down, exactly. You need to keep them both in the same line. And how do you know that you have that kind of level? Because this is you assuming, right? Because you can't actually pinpoint the levels that you have. They change, I think, that every day. Because your mood and your reactions to your environment are not always the same, right? So how you respond to your environment also changes your levels, and your diet also changes your levels. So it's a very delicate equilibrium. And yeah, it's not something that you can pinpoint every single day. It's not like diabetes, that you take out blood and there is an indicator that has already been established, and you can regulate that in between, you know? And so,
Yo pensaría, bueno, no sé, en mi opinión, que quizás sí puede haber una variación también, pero dependiendo cómo se encuentre la persona, porque entiendo que los pacientes que son diagnosticados con este tipo de problemas son medicados, y pues se supone que también hay alguien que tiene que estar pendiente, porque como se menciona, es un tipo de discapacidad. Ok, la persona se ve normal, la tenemos pues de forma bastante normal, podemos hablar y pues ellas pueden hacer su vida siempre y cuando pues estén controlando la parte de los medicamentos. Eh, Entiendo que al suspenderlo pues empieza una serie de episodios que van pues progresando con el tiempo, y ahí es donde pensaría lo que estás mencionando con respecto a que cambian muchas cosas pues con su dieta, porque ya no quieren comer, o ellos comen pues como que lo que quieren en el momento, y puede alterar el organismo como tal. No sé si de pronto eso también juega un papel importante.
definitivamente sí.
Porque entiendo que básicamente mi pregunta sería, bueno, en este caso una preguntita que tendría es, si un paciente, pues asumamos que él esté súper juicioso, o ella, no sé, o sea, esté súper juicioso, todo el tiempo se controle, o hay alguien que esté pendiente y vele porque esté controlándose, ¿habría algún factor que en algún momento genere que caiga o en una crisis, digamos, de este tipo de
Sí.
enfermedad? Pero sería
Dentro de
más,
la misma enfermedad, ¿tú te refieres?
o sea, sí, o sea, digamos que la persona se está controlando médicamente, que es la forma que entiendo que puede ayudarse a estas personas a,
Ujú.
a estar, pues, lo más equilibrado posible. Pero habría algún, no sé, algún factor dentro de la misma enfermedad o dentro del mismo que haga que, a pesar de que esté medicado, pues como que ocurra un episodio de crisis, o que por lo menos empiece a presentar eh, síntomas de que como puede llegar a una crisis, o eso ya depende pues de otros factores que no podemos explicarlos hasta el momento.
Mira, un caso bien sencillo es como la depresión, es parte del diagnóstico, ¿no? Un caso bien sencillo es que se le muere un familiar que la persona ame mucho y esa noticia le venga de repente, eso no le va a sentar bien a la persona. Entonces hay que estar bien pendiente a la persona porque va a caer en una tristeza, obviamente, y esa tristeza pues puede ser la depresión reactiva, ¿no? Que es momentánea,
Okay.
pero esa depresión se puede convertir en un episodio grande de depresión. ¿Ves?
Entiendo. Sí, sí.
Entonces, ahí empieza
It's not so easy to pinpoint because even the lithium response, there's even some lines of thinking that bipolar disease, these two might actually be two different diseases that have overlapping physiological effects. So it's not so easy. So just sort of as a segue to the article that you guys read, that was one of the reasons why the authors of that work, they wanted to see, well, what is it about lithium that people are responding to? Because obviously lithium is limited in its capacity to help people. So the idea was, well, what is it that lithium is regulating or potentially contributing to regulation that might lead to an insight as to a specific molecular pathway that would require drugging, and that lithium is sort of indirectly hitting at, but a better drug might be able to more directly have an impact. So that became the subject of that article, looking at the lithium response pathway. And I saw, just to briefly look at some of the things that people posted. So I saw Omar had an issue with the writing of these articles. So yeah, scientific writing can be a little bit grandeur, and so simplicity is much needed. I would say some of it also has to do with the techniques that you really can't simplify in terms of making it easy for everyone to digest. It's kind of you got to look it up and make sense of it for yourself, because there's certain things that are particular to a field that it's just hard to break down every single time that you need to talk about something. But so trying to really get an understanding, so this was the focus of this paper. What is it that lithium is impacting on certain patients of this condition that might lead to new insight, new drugs? And so I'm going to skip over this because we're going to look at it in our next lecture. So we're not going to finish our discussion today, and that's perfectly fine, because I wanted to just emphasize a few things. So again, highlighting the objectives of this particular study. So looking at examining lithium's drug target in lithium-responsive bipolar disorder for a molecular insight into the actual disease. Because most of lithium's actions have been linked to post-translational regulation of proteins rather than to transcription, that is what gave these authors an idea to, well, what if we take a proteomics approach to try to understand potential protein level differences that are correlated with lithium treatment. So post-translational means that after a protein is expressed, it gets processed, and that processing can afford certain functionality. And then the actual work commences with human-induced pluripotent stem cell-derived neurons, then continues with mechanistic insight validation in mice models, and then also looking at certain human brain specimens to try to get more of an insight of the correlations that this manuscript tries to establish with lithium. So there, in terms of breaking down this work, you can break it down to the component of them simply doing a proteomics type study. And that is just to understand what proteins are getting affected by these neurons due to a lithium treatment. And so there was a specific type of mass spectrometry experiment that was done to gauge the proteins that are getting affected. And then coupling that proteomic study with bioinformatics to try to identify specific pathways that these proteins are correlated with. And so you will see, as part of this work, that they do find a connection with that GSK3B enzyme that I pointed to, and that also gets somebody highlighted, that's correlated with lithium. And they do a neurocytological and behavioral analysis of transgenic mice, in which an important pathway is identified by proteomics. And so they try to really look at this correlation between what was established with the neurons to what's observed in these mice. and then to compare the findings with biochemical and histological assessment of primary human patient brain specimens. Because again, what you observe with animal models may not necessarily correlate with humans, so that they wanted to really look at it from the level of looking at neurons, then translating it to animal studies, and then translating it to actual human sample. All right, so essentially their starting point was by working on these human-induced pluripotent stem cell neurons, and they were looking at what happens with lithium responsive and lithium non-responsive bipolar disorder, and also unaffected individuals. And they also focused on a patient with Parkinson's disease, just to have a control of someone with or a condition that's neurologically affected, but that is not necessarily correlated with bipolar disorder. So, but they observed no differences in neuronal induction efficiency and yield from all subjects. So you had these similar profiles, similar expression of neuronal markers. You produce your neuroprogenitor cells. So this is a transition from the stem cells to these type of cells. And so what was done was an unbiased mass spectrometry-based proteomic study. So here you have just like an overview, and I'm not sure why it looks blurry, but anyways, basically taking these neurons, treating them with lithium, and then also obviously as a control, not treating them with lithium, and then doing a two-dimensional gel to try to separate the proteins, and then getting those different proteins that are isolated in the gel and running a mass spectrometry experiment to identify the protein levels differences between the treated and untreated samples. And so here, what was observed were that there were 26 differentially represented protein spots, which correlated with 15 distinct proteins. All right, so that was what was obtained from this actual study. And so that running that collection of data against a human gene expression database, right, they were then able to look at certain key proteins or genes, excuse me, that increase in response to the lithium, and that were differentially expressed in the bipolar disorder compared to the control brains. And so these were the three that were identified: the collapsing response mediator protein 2, so CRMP2, the mitochondrial ribosomal protein S22, and cystatin. So what they did was then to correlate this work, again, with some bioinformatics studies. So they did some analysis of the Sullivan Lab Evidence Project, and to indicate proteins whose encoding genes might be linked to psychiatric disorders. And they also, from that analysis, identified CRMP2, which was also identified in the mass spectrometry study. So that particular CRMP2 is the only candidate protein to be encoded within the chromosomal region 8p22-21. These are linked genes or risk factors for psychiatric disease. So this became something that they highlighted, and they wanted to look at more closely to see, well, what exactly is the lithium impacting on? All right, so and ingenuity pathway analysis and string network tool modeling. So again, a bioinformatics approach to try to really get some correlations. You want to identify pathways that are being impacted on from a identification of a particular gene or protein. And so here's what they observed was that lithium treatment of human neurons appears to modulate 4 pathways, of which all 4 of these pathways, the CRMP2 is correlated. So here, I'm not going to actually name them, but these are the 4 pathways that were identified from this study. All right. So what about CRMP2? As it is involved in axonal guidance and cytoskeletal dynamics of the neurons. The phosphorylation is actually a very key phosphorylation event of that protein, which occurs at the amino acid threonine 514. So that phosphorylation event appears to be very important, and it's facilitated by kinases, which causes its dissociation from cytoskeletal proteins. So it's a precursor required phosphorylation event that takes place at the S522 amino acid. So the IPA analysis showed that the lithium responsive pathway in bipolar disorder neurons overlaps with the enzyme GSK3b. So there's something that's correlating GSK3b and the CRMP2. Okay, so what is this correlation? What's going on here? And what impact is the lithium having in this? All right, so the question is, and so let me actually clear that up so you guys can see what I wrote there. Is the CRMP2 the primary GSK3b substrate in bipolar disorder high PSC neurons? So that was the guiding question, okay? And so what they did was additional mass spectrometry experiments to look at what happens when you use an inhibitor of the GSK3b enzyme. And so we're going to look at this, and we're going to end our discussion for today with this slide, where using that inhibitor, what they were able to see was the following. So we look at this response, the dose response of the inhibitor, and what you're seeing is that as you increase the amount of inhibitor of that enzyme, you see that there is a decrease in the phosphorylation of the CRMP2 protein. So that phosphorylation at the T514. But when you compare the total levels of the protein itself, okay, the dose response, there actually is no significant difference. You see what trends to be a decrease, but if you do the statistics, that amount of decrease is not statistically significant. So it isn't that the protein level of the CRMP2 is getting impacted on. It is the phosphorylation of that protein that's getting affected, and that may be correlated with the lithium. And so here, they examine lithium contribution to the phosphorregulation of CRMP2 in the high PSC-derived neurons. So you have a series of experiments that look at what's happening due to the presence of the lithium. And so here, it was focused on this gel to the right, where you look at the response based on the presence of lithium and other metals, sodium, magnesium 2+. And what you can see is that when you have the presence of lithium, that actually is correlated with a decrease in the phosphorylation of the CRMP2 and protein. And that's also observed even with the co-treatments with sodium and magnesium, where the sodium and the magnesium by themselves do not have that effect. So it appears that the lithium is somehow regulating that phosphorylation. All right, so I'm going to actually end there, and we're going to pick up from here so we can see what is believed to be the further correlations of the lithium in terms of this process, and how that might actually give potential new insight into druggability for this condition. So I want to thank Getsamari for her insight, giving us a perspective as to the significance of this problem, and then allowing us to see like possible ways that we have to consider, the complexity of the disease and really the control, not just in this case of a metal that may contribute to, trying to improve the disease, but just really how many factors are involved in this particular disease. All right, thank you. All right, everyone. So I will see you next time, and we'll continue on from here. Take care.
Professor, sorry,
Yeah.
I think that, no, it's me, Getsamari.
Yeah. Go ahead.
I thought that, I can't recall if I stayed on time or not.
Oh, you're fine. Thank you.
I think that, but it's actually one of the, I was reading the chat, and some